Hello students, this is Ashta K. Sanush, Assistant Professor, Department of Microbiology, EMA College of Arts and Science. This is a continuation of our web video series that is genetic basis of antibody diversity. Okay, let's get started. Let me choose a pen. Here we go. So we have seen in our previous videos that antibody generated by the production of heavy chain and light chain polypeptides that are synthesized from Ig genes, right? So antibodies are actually generated by the immunoglobulin genes. Now, if you see the heavy chain gene locus, which we have already mentioned in a gene organization video, so it is present on chromosome number, let me just write it in short, chromosome number 14 in the case of humans, and in the case of mice, it is 12. And for light chain, it is on kappa, okay? Kappa is present on chromosome number 2 in the case of human, and in the case of mice, it is present in chromosome number 6. And lambda, it is present in the chromosome number 22 in the case of human, and 16 in the case of mice. We have already seen that the immunoglobulin gene is comprised of variable V region, in diversity D region, then joining J region in the case of heavy chain. And in the case of light chain, we have variable V region and joining J region. Okay. Along with this, we have a constant C region present. Now, this constant region, we know, will determine, that is present in the heavy chain, will determine whether the antibody will be IgM, IgG, IgE, IgA, or IgD. Let me write it here. Okay. Now, we need a combination of this VDJ, okay, to occur, plus we need the V and J of the light chain to occur for the antibody diversity. And we mentioned about the 3 Ig loci. That is for heavy chain, it is 1. In the case of light chain, it is 2. So our 2 of each allele are represented in the genome. So only one HC or LC and not or and LC gene will be expressed, that is the Ig gene will be activated. The rest will be silenced. Okay, so I think I mentioned it in my previous video where I mentioned that the genes expressed in our body comes from mother or father. So the B cell, single B cell can have a gene coming from mother. Okay, it can get gene from mother or from the father. Okay, it is all from the father, but not from both. So that doesn't mean that all the B cells, if particularly this B cell is actually expressing the mother gene and it is silencing the father gene here. But that doesn't mean that rest of the cells or rest of the B cells will take up only M genes coming from mother genes. Okay, it is just one cell. It is going to take mother, or it is going to express the mother gene, and the silenced one in that case is the father gene. Maybe the other cell might express the father, maybe the other mother. It depends each individual cells, right? I'm talking about each individual B cells can take up either genes from mother or from the father, but not from both. So what happens when it is taking up genes from one alleles, it is silencing the other allele. So this whole process is called as allelic exclusion. We also know that the combination of VDJ and VJ, it is required, only then the genes can become functional. So it can become functional gene. So actually you can say it in this way, that the immunoglobulin genes which are found in the genome are not functional, and that they need to be activated to produce, okay, to produce the antibodies. Okay, now the process is initiated in the bone marrow. It is initiated in the bone marrow at the pro B cell stage. If the rearrangement is faulty, then one of the other light chain Ig alleles can compensate for the defective process. So you actually, I've just mentioned it, there are only one allele for B cell and three allele in the case, for heavy chain, and three allele left for the light chain. Suppose the one which has been chosen, it is faulty. Okay, so in that case, they can opt for the other allele which is present, and they can, which can actually help or compensate for the defective process, right? So otherwise, if what happens, if both the alleles in the case of heavy chain is faulty, it will undergo a process known as apoptosis. And same thing actually occurs for light chain also, where if the all the possibility alleles are faulty, it will undergo a process named as apoptosis. Okay, so light chain do have a choice with three other alleles, and heavy chain just has one. Once we have a functional heavy chain gene, and then you have the functional light chain genes, now the cell will become mature, and the cell becomes immature B cells, and it will start migrating from the bone marrow to the spleen for further maturation. Okay, now let me just give you a gist of what actually happens, a brief, and we will see in next series the whole thing, like what actually happens in this VDJ recombinations. Okay. Yes. Now, as I told you, this is the germ line. Suppose you have a region for V, then you have a region for D, and then you have a region for J, then it is followed by the constant. Yeah. This is the V, and this can be M, D, E, A, G. Okay. So what happens, as I told you, in the gene organization, there will be certain, like, let me just write it down here. I think that's will be better. In the case of heavy chain, the variable in the case of human, it will be around 40 variable regions in the case of heavy chain. And in the case of D, okay, D, it might be around 24. In the case of J, that is the joining region, it will have a gene pool of 6, right? So you have 40, okay, so it will be there. Just consider if it is 40 V1, V2, Vn, okay? Then you have 24 here, D1, D2, then I'll just write it Dn, okay, J1, J2, Jn, okay, followed by the constant. So initially in certain books, they have shown the V and J recombination occurs. In certain books, the recombination of D and J first occurs. So let me just follow the book which I actually referred. So one part of B and one part of J, okay, one part of each is going to join, okay. So one part, in the sense, maybe it has chosen randomly D2 and maybe DJ1, okay? So it gets joined. So now at this stage, you can say it is DG, recombination. And after that, one part of variable B region also comes and bind. Okay, this is the first step, and then this comes and joins. Now this one part from each has combined. Okay, along with this, you'll have the constant region also, right? So you have the, we just write it a VDJ coming together after the recombination, followed by, you have the constant. Okay, let me just write it like constant region. Okay. Now what happens from here? It gets transcribed. Okay. Now it will have B, D, and J, followed by the constant. Okay. This is the mRNA, right? After which splicing occurs. So after splicing, whatever the constant, may be this part or this, okay, gets chosen, and it also combines with the VDJ. Suppose here the expressed is M. Okay, let me do this. VDJ and M. Okay. Now what will happen? This will again undergo translation, and the proteins formed will help in the, proteins formed are for heavy chain. This is a brief explanation of this PDJ recombination, and next video I'll be telling you about in detail we'll be talking about what actually happens, or we are going to zoom in into what actually happens in this recombination. So similarly, if you take the same process happens for light chain also, where you have V and J region alone. So one part of both of this will come and bind, so I don't have to repeat it. So this is going to come, and for the process of transcription, then it gets sliced. Okay, so after which it is going to form the proteins, which is for light chain. Okay, now in the next video, I'll be explaining you in detail of the process. Thank you.